Hello and welcome to Stories of Science. Here, we look back on humanity's long journey to understand the world. The questions, the failures, and the breakthroughs that slowly changed how we see the universe and ourselves. We tell science as stories. The scientist who saw the future, how one man's vision of DNA changed the world. If one day, you could read DNA, not as a doctor, not as a scientist, but as yourself, you open a sequence. It's like reading a book. On those pages, you'll find your talents, your diseases, even your lifespan. Sounds like science fiction, right? But the first person to get close to this ability? Not a biologist, a physicist, someone studying particles, energy, things far from life. But one day, he turned. From stars, he jumped into cells. From the universe, he dove into DNA. That man was Walter Gilbert, an unlikely hero. And what he did, it shook the world of science. Back then, reading even a small piece of DNA seemed impossible. Experiments were slow, the data scarce. No one believed humans could decode life. But Gilbert said, "We can. Soon." In 1986, he said it like a prophecy. Humans will read the entire genome. People probably laughed. But why did he believe this? What did he see? Let's go back to 1932. A boy was born in Boston. No lightning, no visions. He seemed ordinary. But his family, not so much. His dad, an economist, but not the type who writes papers, the type who shapes policy. His mom, a child psychologist. She studied how kids' brains worked, while other kids grew up. He was the subject of research. Imagine that. Instead of being raised, he was tested. While others played with mud, he did cognitive experiments. While others were asked, "Are you happy?" he was asked, "How's your logic?" Even his emotions? Data. This wasn't a home. It was a mini-lab, a tiny research institute. And he was the only subject. In 1939, at 7 years old, they moved to Washington. The environment changed. But one thing stayed the same. He was always observed. But slowly, something strange happened. He got used to it. He even started to enjoy it. Analyzing, thinking, solving problems. For him, it wasn't work. It was instinct. Was he a genius? Or just well-trained? Either way, he was different. And this different would change science. Fast forward to 1950. At 18, he entered Harvard, majoring in physics. No struggle. Everything went smoothly. In 1953, he got his degree. And his goal? Higher, harder, more abstract. So, he went to Cambridge. Cambridge, the mecca of theoretical physics. The air thick with equations. At Cambridge, he met Abdus Salam, a future Nobel laureate. Gilbert, a student of particle physics. Sounds uninviting, right? Invisible, intangible, all math and imagination. They were trying to understand the deepest rules of the universe, the smallest particles, the most basic interactions. In other words, they were asking, why does the world exist? Poetic, right? But the reality? Long hours, blackboards filled with equations, no applause, no audience, just endless calculations. By 1957, he had his PhD, perfect resume, standard path, from Boston to Washington, Harvard to Cambridge, on to cutting-edge physics. Seems like a standard success story, right? Great, stable. But the real story? That was about to begin. In 1960, his life was still on track. Physics, equations, particles. But then one man appeared. James Watson. The same Watson who, in 1953, cracked DNA's structure. The double helix. Elegant. But back then, it was like opening a door to the unknown. Watson invited people to his lab. No commitment. No plan. Just come, look, talk. Sometimes, life changes happen in the most casual moments. Gilbert walked into the lab, and instantly, everything felt different. No blackboards filled with equations. No invisible particles. There were test tubes. Solutions. Life happening in real time. But more than that. There was a question. A simple question. How does DNA turn into protein? Seems basic, right? But no one knew. DNA was the blueprint. Proteins were the workers. But the process? A mystery. And that question hit Gilbert hard. It wasn't just interesting. It was a revelation. Like realizing you've been studying the surface. And the real mystery is deeper. From that moment, his path began to change. No longer just particles. Now, he was drawn to life's core. DNA. A molecule smaller than a particle. But maybe more powerful than the whole universe. It sounded absurd. Like an astronomer studying stars. Suddenly turning to cooking. But to Gilbert, it wasn't a leap. It was inevitable. The question was the same. What is the structure? What are the rules? How does information flow? The stage changed. From the universe to the cell. From infinite vastness to tiny detail. And from that moment on, things could never go back to the way they were. Gilbert didn't quit right away. Everything seemed normal. But inside, his trajectory had shifted. You could keep working in your old field, but now you'd seen the bigger question. Like seeing the ocean. You could never be satisfied with a puddle. Slowly, he moved toward biology. Reading, learning, understanding that life was messier than physics, but also more real, more dangerous, more likely to change the world. In 1960, nothing major happened, but his life shifted completely. He wasn't just a physicist anymore. He was an outsider. And he was about to enter the heart of life. After 1960, Gilbert did something others thought was crazy. He jumped from physics into molecular biology. It wasn't just a subject change. It was a whole new worldview. Physics was clean, elegant. Biology? Messy, noisy, variables everywhere. You ask a question, and the answer is, it depends. For a physicist, that's almost torturous. But Gilbert did it, and never looked back. He learned a new language. Bases, nucleic acids, enzymes, words that felt alive. Nothing like the calm formulas of physics. They were characters. You thought they'd follow rules, but they'd flip at any moment. At that time, biology stood at a crossroads. A new idea was emerging. The central dogma. DNA, RNA, protein. Simple, clean. But the problem? No one knew how it worked. Sure, the path was there, but the details were a blur. How does DNA turn into RNA? How does RNA guide protein? Every step was a mystery, like getting a manual with a title user guide. But the pages were just gibberish. You knew it was important, but you couldn't understand it. And Gilbert? He loved this challenge. He was hooked. He started researching mRNA. Messenger RNA. It delivers DNA's message. But how does it form? How does it copy information accurately? No one knew. And the experiments were tough. A lot of guessing. Guess right? Genius. Guess wrong? Try again. But Gilbert had a physicist's mind. He asked, what's the mechanism? Where's the process? How do we control the variables? At the time, biology was stuck in the descriptive stage. Gilbert wanted explanations. Real explanations. By the mid-1960s, Gilbert was becoming a force in biology. Not because he knew everything, but because he was willing to admit no one knew. That became his advantage. He didn't have the burden of tradition. He could start fresh, reframe questions, redefine paths. But the road was tough. He wasn't a traditional biologist, nor was he purely a physicist. Stuck in between. A little awkward. But that's what gave him the ability to see things others couldn't. From 1960 to 1970, no explosive breakthroughs, no fame, just trial and error, getting closer to the truth. And the question remained, how does DNA talk? How does it turn information into real, molecular machines? The field was still searching, still trying to read the book. But Gilbert, He was becoming the one who could finally read it. It's the late 1960s now, and the air is changing. No longer just confusion, but a sense of maybe we're starting to understand. But the problems? They're sharper. If DNA is the instruction manual, then why are some genes working overtime while others seem to be slacking off? The same DNA, in different cells, behaving completely differently. One cell is busy making proteins, while another is just resting. Something doesn't add up. The manual should be the same, but the results are all over the place. And then, a bold idea emerges. What if genes have switches? Not always on, not on autopilot, but controllable. Pause when needed, turn on when it's time to go. This idea? It sounded almost like science fiction back then, but others were already ahead of the curve. François Jacob, Jacques Monod. They were studying something simple. E. coli. Tiny, simple, yet remarkably smart. They found something fascinating. When lactose is in the environment, the bacteria flip a genetic switch. They start breaking down lactose. When lactose is gone, the genes turn off. No energy wasted. It's like an accountant meticulously keeping track of every penny. They came up with a model, the lac operon. Sounds complicated? Actually, it's pretty down to earth. It's a turn on when needed system, on when necessary, off when not. And just like that, a key conclusion: genes aren't always working like machines. They're part of a managed system. But there's a new question. Who's doing the managing? Who decides when to flip the switch? We've got the theory. We've got the model. But the switch? Still missing. It's like the light can turn on, but you don't know where the switch is. The whole field was stuck. And then, he decided to take matters into his own hands. Not more guessing. Not more models. He was going to find that switch and pull it straight out of the cell. Sounds simple, right? But back then, it was like asking for trouble. You're not dealing with a clear target. You're facing a molecular mess. Cells are broken open. Inside, thousands, maybe even millions, of proteins, all mixed up. And you're looking for just one, with no label, no color, no hints. It's like looking for a single drop of water in the ocean. This wasn't an experiment. It was a test of patience. He tried. Again. And again. Extracting. Separating. Then separating again. Trying every method to make the target protein pop up. Failing? That was the norm. Success? That was the occasional illusion. Sometimes, you thought you found it. Then, the next day, you realize it wasn't right. Back to square one. There are no miracles in the lab. Just repetition. And countless almost there moments. But he had one advantage. A physicist's mindset. He wasn't satisfied with close enough. He wanted certainty. Mechanisms. Repeatable results. And finally, in the late 1960s, it showed up. The lac repressor. Sounds cold, right? Like a tool. But its significance was nothing short of revolutionary. It was the switch. When it's present, it binds tightly to the DNA and stops gene expression, like a guard standing at the door, keeping everyone out. But when lactose shows up, the repressor is released. The door opens. Genes start to work. Proteins are made. Clean. Efficient. Like flipping a switch. For the first time, humans didn't just guess. They saw it. Genes can be turned off. Genes can be turned on. Life isn't a never-ending conveyor belt. It's a smart system. With conditions, decisions, control. In simple terms, genes aren't on autoplay. They have a pause button, and even a play only when needed mode. That changed everything. Biology leveled up. From observing phenomena to understanding mechanisms. From describing life to controlling life. Because once you know where the switch is, you can influence it, adjust it, even rewrite it. And that's exactly what he did. He turned an abstract model into a tangible molecule. He took maybe it exists, and turned it into it's right here. It seemed like just a protein, but in reality, it opened the door to genetic engineering. to biotechnology, and eventually, to the future of medicine. What's most fascinating? This all came from someone who wasn't a traditional biologist, but someone who had once studied the universe. He moved from the grandest scale to the smallest level, and found the switch that controls life in a cell. Things were about to get even crazier. It's the 1970s now. The stage has shifted, and the problems have leveled up. It's no longer just about how genes turn on and off. Now, there's a more direct question. Can we read DNA? Not just guess, not infer, but read it, word by word, like reading a book. Seems logical, right? But back then, it was nearly impossible. Because DNA isn't written in letters. It's written in 4 bases: A, T, C, G, combined in patterns so long, humans couldn't even read a few dozen bases, let alone an entire gene, or the whole genome. But still, some people started trying, and not just one person. A quiet race to decode life began. On one side, there was him, the outsider, coming from physics. On the other side was Frederick Sanger, a cool-headed experimental genius. Both had the same goal: to read DNA. But their paths? Completely different. It wasn't just competition. It was more like a battle of philosophies. One leaning toward chemical breakdowns, the other towards enzymatic replication. No one knew which way would be faster. In fact, no one was even sure if there was a way. In 1973, a small breakthrough. He successfully read a DNA sequence, 24 bases long. Today, it seems laughable. 24 bases? You couldn't even form a sentence. But back then, this was the first time humanity had really read DNA. Not inferred, not guessed. A confirmed sequence. A, T, G, C, one after another. Like cracking a code. Like finally understanding a word in an alien language. Short, yes, but monumental. It made everyone realize one thing: this isn't impossible. It's just slow. So slow, in fact, that reading a small piece of DNA could take months, maybe longer. At this rate, reading the entire genome would take centuries. No one had the patience for that, and no one had the budget. So the question changed. Not can we read it, but how can we read it faster? And that's when the real race began. He started developing a method, using chemistry, specific reagents to cut DNA at different points, then analyzing those fragments to figure out the sequence. It sounds complex, but it's really just breaking it up, then piecing it together. On the other side, Sanger took a different approach, using enzymes to replicate DNA. But at key points, he'd stop the replication. That way, you get different lengths of copies. And from there, read the sequence. One was about breaking it down, the other about building it up. It was like disarming a bomb versus assembling a model. They were both doing the same thing, but from entirely different worlds. In 1977, the key year, his method? It was ready. And soon after, Sanger's method was ready too. 2 methods, 2 breakthroughs, born almost simultaneously. 2 parallel timelines, both leading to the same future. At that moment, humanity had the ability to really read life. Not guess, not infer, but directly read. DNA. From mystery to information. It could be recorded, analyzed, even archived. What's the significance of this? It's hard to capture in one sentence, because it didn't just change one field, it changed how we think about everything. From that moment on, life was no longer just something living, it was a code, one that could be read, understood, and even edited. And the race? It's like 2 people cracking an alien code. Who will get there first? Who's got the better method? At the time, no one knew. But one thing was clear. They weren't writing papers. They were translating a manual written in every cell. A manual that's been copied since the dawn of life. A manual that doesn't speak human languages, but is more important than any language we've ever known. The most fascinating part? This book isn't owned by one person or one country. It's shared by all life. You, me, a tree, a bacterium. We're all using the same alphabet, just arranged differently, with different stories. After 1980, everything accelerated. Sequencing technology improved. Faster, cheaper. What used to take months now takes days. Scientists no longer satisfied with reading short stretches of DNA. They wanted to read entire genes, even whole genomes. That bold idea from 1986? It started becoming possible. And the race? It started to have a winner. Sanger's method? More stable, easier to use. It became the mainstream method. Gilbert's? It faded into the background. But that didn't matter. He did something even more fundamental. He proved that DNA could be systematically read. It was like the first airplane. It didn't fly far, and it wasn't practical. But it answered one question: can humans fly? The answer: yes, we can. From that moment on, technology would improve, methods would optimize, speeds would increase. But the direction? It was set. So when people look back at 1980, they'll see not just an award, but a beginning. For the first time, humanity had the ability to read its own instruction manual. And the story? Far from over. By 1986, the story was, by all accounts, pretty much wrapped up. Nobel Prize? Check. Methods figured out? Check. His place in history was secure. Now, most people would have done one of two things: sit back, enjoy the rewards, maybe write a few papers, mentor students, and keep doing what they've been doing. But not him. He didn't take the typical route. He went even bigger, bolder, and, quite frankly, a little scary. He proposed something wild. We should sequence the entire human genome. Hold on. This wasn't just sequencing a segment or a single gene. No, this was every single base pair. All 3 billion of them. Not a single one left out. Now, if you're hearing this now, it probably sounds like a reasonable idea. But in 1986, that suggestion was as outlandish as saying, we should transcribe the entire internet by hand. The technology back then? It was painfully slow. You'd spend ages sequencing just a tiny bit of DNA. The equipment was sluggish. The process was complicated. Data analysis? Primitive. And don't even get started on the costs, it was off the charts. So let's do the math. If you wanted to sequence the entire human genome, with the tools they had back then, you'd need over a century. And that's if it went perfectly. With the budget of a small country. And even then, it might not work. When he proposed this idea, the reaction in the room? Mixed. Some were quiet. Others frowned. And a few, they laughed. Not in a mean way, but in that is he out of his mind kind of laugh. Let me translate the unspoken thoughts from back then. Dude, you just read a few hundred bases. Now you want to sequence 3 billion. Are you even awake right now? It wasn't about being cautious. It was just plain reality. The tech wasn't there. But he knew that. He wasn't naive. He understood the costs. The challenges. He simply saw a trend: sequencing would get faster. It would get cheaper. Just like computers and chips. At first, they were slow and expensive, and no one used them. But once the breakthrough happens, they scale up exponentially. He wasn't focused on whether it was doable right now. He was thinking, this is going to happen. It's just a matter of time. So he proposed the plan not to do it immediately but to set a goal. To give the entire field a direction. It's like standing at the base of a mountain. You know you won't climb it today, but you have to decide if you're going to try. And he pointed to the highest peak. The full human genome. All 3 billion bases. An insanely ambitious goal. Over time, the idea started to catch on. Some opposed it, thinking it was a waste of resources. Others backed it, saying it was the future. Debates raged. But something shifted. People stopped asking, should we sequence it? And started asking, when can we finish it? That was the turning point. From impossible, to just a matter of time. Standing in 1986, he could already see the world of 20 years ahead. Back then, there were no high-speed sequencers, no supercomputers, not even widespread internet. But he was already thinking, what will happen when we've sequenced the entire human genome? Medicine would change. Diseases would be redefined. Individual differences would be quantified. Even the very concept of human would shift. Of course, most people weren't quite there yet. They were still wrestling with a more practical question. Can we even pull this off? The answer? In those days, it was still uncertain. But sometimes, scientific progress doesn't start with certainty. It starts with an idea that sounds a little crazy. In 1986, when he voiced this idea, a lot of people thought he was dreaming. But history would prove that he wasn't dreaming at all. He was just waking up before everyone else. Some scientists prefer to stay quietly in their labs. Others, like Walter Gilbert, aren't content to just settle. By 1978, he had already sparked a revolution in DNA sequencing. He wrote the papers. The Nobel was on its way. What would most people do next? Relax. Collect their award. Maybe write a memoir. But not him. He did something even more outrageous. He walked out of the lab and decided to start a company. Yes, from studying life to commercializing life. That same year, he helped found Biogen, one of the world's first biotechnology companies. Remember, biotech company wasn't even a fully coined term yet. Most people hearing about it probably reacted like this. You're going to make money off DNA. Sounds like science fiction, right? Maybe even a scam. But Gilbert had a clear vision. He'd already realized one thing. DNA wasn't just a scientific issue. It was information. And information could be read, stored, and even commercialized. In other words, the future would involve selling genes. It might sound like something out of Black Mirror, but in 1978, it was more of a prediction. Biogen had a clear goal: turn basic research into medicine, take molecules from the lab, and turn them into real-world treatments. At the time, most scientists were content with writing papers. But Gilbert, he wanted to turn those papers into products. That wasn't easy. Success in the lab didn't guarantee success in the real world. In the lab, you control everything. But in the real world, you don't control the market. You don't control capital. You definitely don't control human fear. This was a time when genetic engineering made a lot of people nervous. Are you going to create monsters? Are you going to mess with humans? It may sound exaggerated, but that's what some people thought back then. Gilbert wasn't just facing scientific challenges. He was up against public opinion, ethical concerns, even political issues. But he didn't back down. In fact, he pushed forward. A few years later, he helped found Myriad Genetics, a company that was even more controversial. They focused on genetic testing. In simple terms, it meant looking at your DNA to see what's written inside. Sounds cool, right? But also a little frightening, because it might tell you if you're at risk for certain diseases, like cancer, or genetic disorders. In other words, it previews your future. Some saw it as a blessing, others, a curse. But what really stirred the pot was Myriad's next move. They began patenting genes. Yes, patents. They treated specific human genes as inventions and applied for ownership. Wait, genes, aren't they natural? Can they really be owned? That was the heart of the controversy. Supporters argued that without patents, there would be no investment. No investment? No technology. Opponents said, you can't turn part of humanity into a product. The debate raged on for decades, and eventually, it went all the way to court. But regardless of which side you were on, one thing was clear. Gilbert had pushed science into a whole new frontier. From discovering natural laws to redefining nature itself. That's a dangerous step, but an incredibly important one, because from that moment on, science wasn't just about understanding the world. It was about changing the world, and doing it with a commercial twist. Interestingly, Gilbert never fully left academia. He kept writing papers, kept participating in scientific discussions. But his role had shifted. He wasn't just a scientist anymore. He was a bridge. One end connected to the lab, the other to the market. Many people didn't like that role. They thought it was impure. But history is often driven by those impure people. They're the ones who dare to cross boundaries, take risks, turn theory into reality, and even into an industry. You could call him pragmatic. You could call him visionary. But you can't deny, he saw a future no one else had yet imagined. While others were still reading DNA, he was already thinking, how do we use DNA to change the world? And even, how do we build an industry around DNA? It might sound capitalistic, but it's also deeply human. Because from fire, to electricity, to the internet, every major breakthrough has gone in one direction: commercialization. And Gilbert? He just got there a little sooner, a little faster, and a little bolder. So, here's the thing. Some people carefully work in labs. Others boldly talk about the future in boardrooms. And Gilbert? He was doing both. Daytime, studying molecules. Nighttime, talking business. While others wrote papers, he was busy launching companies. Sounds like a punchline, right? But it was real. And the most interesting part? This was just the beginning. A lot of people think DNA sequencing is just a lab story, something far removed from their lives, like a world seen through a microscope. You see it, but can't use it. But the reality? It's the complete opposite. Today, a lot of the medical tests you do, they're tied to one name, Walter Gilbert. And you've probably already used his work, even if you didn't know it. Take genetic testing. Sounds fancy, right? But it's becoming more and more routine. All you need is a blood sample, or a cotton swab, and the lab can read part of your DNA, tell you your risk for certain diseases, how you might react to certain medications, even where your ancestors came from. The logic behind it is simple. DNA is just a code. You read it line by line, analyze it segment by segment. And before the 1970s, this was nearly impossible. It was Gilbert's generation that turned reading DNA into a real technology. And cancer screening? It used to be that cancer wasn't caught until it was too late. When symptoms showed up, it was often already serious. Now, doctors can spot risks earlier by looking for genetic mutations, like the BRCA gene. If it mutates, your risk for breast cancer skyrockets. This isn't a prediction, it's data. And this ability to warn the future with DNA? It all started with sequencing. And let's go one step further. Personalized medicine. Sounds like sci-fi? But it's already happening. The same drug might work wonders for one person, but cause serious side effects for someone else. Why? Because of their DNA. Some people metabolize it faster, some slower, some respond well, others, not so much. So doctors are starting to do something. First, let's look at your genes, and then we'll prescribe the medicine. Sounds like tailor-made treatment, right? Yep. Medicine is becoming customized. And the foundation for all of this? It's reading DNA. From those initial 24 bases to today's millions of data points. The speed has increased many times over, and the cost has dropped many times over. It used to take years just to sequence a gene. Now, sequencing an entire genome could take just days, maybe even faster. And all of this? It started decades ago, with those few people in the lab, translating the instruction manual of life. You might not have seen their faces, but that line in your medical report about DNA? That's their legacy. You might think you're just reading a report, but really, you're reading history. History that starts at the molecular level and stretches all the way to life itself. The funny part? Gilbert started out as a physicist. He was studying the laws of the universe. Then he shifted to biology, studying the code of life. Seems like a whole new field, right? But really, he didn't change. He just changed the scale of the problem. From galaxies to cells, from the structure of the universe to the structure of life. At its core, it's all the same question: how is the world written? Physics answers it with equations, biology answers it with DNA, and he happened to stand right between them. Some people spend their lives studying the world, some spend their lives explaining it, but then, there are the special ones. They don't just want to see, they want to read. They treat the world like a book, flipping through pages, line by line, from the universe to life, from energy to information. And that's where the story truly starts to become clear. Some people study the world, others, they read it. The people who change the world usually just think one step deeper. If this story made you see things differently, hit like, subscribe, and save it. See you in the next story.